Okay, let's dive right into this. Welcome to today's Explainer, where we're taking a really close look at a totally groundbreaking new path in medical research. Today, we're focusing on what is basically a devastating and honestly often unseen enemy that affects families in just profound, totally life-altering ways. But more importantly, we're looking at the incredible scientific journey happening right now to finally crack its code.
I mean, think about it. How do you fight a disease you can't really see? It sounds kind of like a riddle, right? But for decades, researchers who've been studying Coates disease have faced exactly this paradox. Coates disease is this rare, highly serious eye condition that mostly strikes kids. What's actually happening is the tiny blood vessels in the retina just fail to develop properly. They get enlarged, they become really unstable, and eventually they literally begin to leak fluid and lipids into the eye. Over time, that leakage leads to massive swelling and tissue damage, and it carries a very real, very scary risk of total vision loss or severe retinal detachment.
So the crucial point here is this tiny, tiny number, 0.1. That represents the maximum number of individuals affected per 100,000 people. It is incredibly rare. And, while you might immediately think, hey, fewer cases, that's a good thing, right? Well, in the world of scientific research, extreme rarity actually slows down progress. It creates this really harsh reality where the urgency of the families affected is just immense, but the scientific tools to study the disease remain heavily limited by those small sample sizes.
And this brings us right to the massive bottleneck in Coates' research. There is currently no widely accepted animal model of Coates disease, and without an animal model, researchers are essentially flying blind. They can't fully observe how the disease develops from its earliest stages, and honestly, more importantly, they can't test potential treatments in a controlled, scientific way to see what actually works. But recently, researchers had a massive aha moment. They noticed another rare eye condition called FRAM, which stands for familial retinal arterial macroaneurysms. This condition shares some very important similarities with Coates disease in how it actually behaves. But here is the real game changer. Unlike Coates disease, which has no known genetic link, FRAM has a very specific known genetic starting point. It's caused by a mutation in a protein called IGF-BP7. I know, it's a mouthful. But that protein directly affects how blood vessels develop.
And that leads us to this incredible revelation. Remember that missing tool we talked about earlier? Get this. It turns out it's been hiding in plain sight the whole time. A mouse model engineered without this protein literally already exists. But until right now, the retinal changes in this specific model have never been fully studied. So this existing model could very well be the exact key needed to finally unlock the biological secrets of Coates disease.
Now, you can't tackle a challenge like this without an absolute dream team, and that is exactly who is leading the charge here. We have Dr. Leo Kim, an incredibly esteemed vitreoretinal surgeon scientist at Massachusetts Eye and Ear and Harvard Medical School. He's working alongside other heavy hitters like Dr. Tom Lee, and the whole shebang is being championed and supported by the Jack McGovern Coates Disease Foundation. The credibility and just the sheer level of expertise converging on this specific challenge is nothing short of remarkable.
Let's move to how this builds out into a precise year one roadmap, because this isn't just a hopeful guess, right? It is highly focused, strategic, and super methodical. Step 1, establish and image the model. Step 2, meticulously track those retinal changes over time. Step 3, carefully analyze whether this model truly reflects the mechanics of Coates disease. And finally, Step 4, begin building a detailed, robust data set that the entire scientific community can actually rely on.
To pull off that roadmap, they are bringing out the heavy technological artillery. They'll be using advanced eye mapping and imaging tools, stuff like OCT, which takes these incredibly detailed cross-section pictures of the retina, and fluorescein angiography, which uses a special dye to literally illuminate the blood vessels so they can spot abnormal changes. But honestly, the real game changer here is artificial intelligence and machine learning. By combining traditional biology with AI, the team can detect incredibly subtle patterns in the imagery that even the most highly trained specialists might just miss with the human eye alone.
Now, I want to pause here for just a second for a moment of complete transparency, because look, in science, honesty is paramount. This is early-stage research. There are no guarantees here, and this specific project is not some magic overnight cure. But what it is the critical first step. Because if it's successful, it creates a validated lab model. And with that model, researchers can finally lay the essential groundwork to identify real, actionable treatment targets for future therapies.
Of course, making this science happen requires clear financial support. To fund this incredibly efficient Year 1 roadmap, the project requires $100,000. But the amazing news, and this brilliantly illustrates the momentum they've already got, $40,000 has already been committed by early supporters. That leaves a very clear, very achievable gap of $60,000 needed to fully fund this first year of breakthrough exploration.
Now, if you're wondering how that funding is actually utilized, it is entirely focused and super efficient. Every single dollar goes directly into the science. It empowers the researchers, it maintains the crucial animal models we just talked about, and it secures the essential lab tools required for those high-tech imaging processes. And it sets the stage perfectly, because based on Year 1's success, the roadmap expands smoothly right into Years 2 and 3 to push those findings even further.
We started this explainer by asking how you fight a disease you can't fully see. Well, the answer is, you finally build the lens to see it. Will you help turn questions into answers for children facing Coates disease?